This video is going to focus on the 5 functions of antibodies. So we've been talking about the humoral response, which involves B cells, which, by the way, get their name because they mature in the bone marrow. And these B cells, remember, once they're activated and they differentiate, some become memory cells and some become plasma cells. And those plasma cells produce these antibodies. The antibodies have this Y shape that you see here. Remember, it's made up of 2 heavy chains, which are in purple, and 2 light chains, which are in green. Now, always pay attention to the details of these figures because they're telling you a lot. Notice how on both the light chains, they're both green, but the top portion is a lighter green. Same thing with the heavy chains. The top portion is a lighter shade of purple. That's because these light shades represent that variable region, and the bottom parts represent the constant region. Remember, what unifies each class of antibody is that they have the same constant region. So all class A antibodies have the same constant region as one another, but class M antibodies have a different constant region, but it's the same amongst all the class M's. Okay? All right, so why is this important? Okay, well, it's important because antibodies have 5 super duper important roles, okay, when trying to fight off an infection. Oh, my computer is spazzing out. Sorry, y'all. Please hold. Huh. Sorry, guys. Hold on, please. Okay, there we go. So one of the first things is going to be neutralization. And I remember this because the antibody is neutralizing the threat. So for example, one threat that an antibody might need to neutralize is an exotoxin. Remember, these are enzymes that can wreak havoc on a cell, and they can actually cause the cell to die. And so this is what would normally happen. Here's the triangular exotoxin. It's going to bind to, maybe get inside the cell, and it's going to damage it. We don't want that to happen. So look at what these antibodies do. Now remember, antibodies that are going after a toxin have their own special name, which is an antitoxin. That's why you see it labeled as such down here. So notice these antitoxins are going to bind to these exotoxins, right? And in doing so, that exotoxin can no longer attach to that host cell. So it's going to save the host cell. So these antibodies have neutralized that exotoxin threat because when they bound to the toxin, that prevented the toxin from being able to bind the host cell. Now, the same thing can happen with a virus. Now, remember, antibodies are normally targeting extracellular pathogens like bacteria, but there are brief moments when a virus is outside of a cell, right? Once it's released from the host, it can be quickly scattering throughout the body until it finds a new target cell to get inside of. So technically, it is possible for an antibody to help neutralize a virus because when the antibody binds to these spike proteins, like you see up here, that means these spike proteins cannot bind to a new host cell. So that means this virus won't be able to get inside of a cell and kill it, okay? So again, the antibody is neutralizing the threat by binding to part of that pathogen or part of the toxin in order to prevent it from binding to a new host cell. Okay, so it's neutralizing the threat. With opsonization, remember, to opsonize means to cover or coat. So the goal is to coat the surface of the pathogen. And so we talked about this with the complement system. We can coat the surface of a pathogen with complement proteins, or we can coat the surface of the pathogen with antibodies, just like you see here. And so this is a picture of a bacteria, we know because it's a bacillus shape. And remember, a lot of bacterial pathogens have a capsule, that sugary sticky layer, which can make it really hard for those phagocytic cells to attach to. And so opsonization is how we fight back against that capsule. By covering that pathogen with antibody, we're essentially giving this phagocytic cell handles for it to grab hold of. Do you see how the phagocytic cell is actually grabbing on to the antibodies? So that's going to allow that pathogen to be phagocytosed and eliminated. The third function is activation of the complement system. So y'all remember, just because we're talking about all these things, inflammation, phagocytosis, complement system, right? B cells, T cells, we talk about them as separate things, but they're all working together to eliminate a pathogen, right? Just like when you watch any type of battle movie, we have people with bows and arrows. We've got a building with high walls, and we've got catapults of things set on fire. We have people with guns, right? We have so many different things happening to try to eliminate that threat. Same thing with our immune response. So just because the complement system is technically part of our second line of defense, it may not always get activated right away. And so remember, there were 3 pathways of the complement system, and any one of them can be activated, but maybe they didn't get activated right away. And so when a complement protein, which is what you see here with C1, the C stands for complement. Hold on, y'all, my computer is passing again. I'm so sorry, I don't know what's going on here. It has never done this. Okay. Sorry, I'm still here. I appreciate your patience. There we go. So this complement protein, when it detects an antibody bound to an antigen, that is going to initiate that complement system cascade, which is going to end up in opsonization, inflammation, and cytolysis. Okay, so remember, we talked about the outcomes of the complement system back in our non-specific immunity videos. And so go back and review that. But again, we're trying to have as many weapons as possible to round up and eliminate this pathogen. So by activating the complement system, even if it's later in the game, that's just more ways more ways for us to get rid of that pathogen. All right, our fourth function of antibodies is immobilization. Remember, a lot of pathogens, so bacteria, some protozoans, which you haven't learned about yet, but a lot of organisms have flagella. And remember, that is used for motility. And so, like the bacteria in this picture, by having all that flagella, they can outrun our immune cells. Well, we don't want that. And so antibodies can attach to that flagella and essentially prevent the flagella from working. So we are immobilizing them so that our immune cells, our phagocytic cells, can come and catch them and eliminate them. All right, the last one is agglutination. We talked about this in lab with a latex agglutination test. So notice which class of antibody we're looking at here. IgM, the pentamer. And this one has a really important role with agglutination because look at what's happening in this picture. This one big old antibody is attached to 2 different pathogens. So it's forcing these pathogens to be in close proximity to one another, right? Same thing with this other antibody. It's got a different pathogen and it's bringing them all close together. So essentially these antibodies are rounding up all the pathogens and bringing them close together. That way the phagocytic cells can actually phagocytose multiple pathogens at one time. Because remember, y'all, when we're sick, there's thousands and thousands and tens of thousands of bacteria in our body that we're trying to eliminate. If we have to wait for one phagocytic cell to phagocytose one single bacteria, we're going to be sick forever. Okay, so by bringing these pathogens together, the phagocytic cells can essentially chow down on multiple pathogens at one time. All right, y'all, that's the end of this video. Let me know if you have any questions.